Hello and welcome to the course Basics of Experimental Design for Cognitive Psychology. I am Dr. Ark Verma from Department of Cognitive Science at IIT Kanpur. This is the 8th week of the course where we are discussing methodologies of experimental research. In this lecture, I will continue talking about neuroimaging methods. Now, we have seen I've given you a broad overview of fMRI and PET in the previous lecture. In today's lecture, I am basically going to touch upon very superficially some of the design questions in fMRI experiments. So, as we know, fMRI measures hemodynamic changes, it does not measure neuronal activity directly, and the signal, the ratio of oxygenated to deoxygenated blood basically varies by physiology. It is a physiological signal, it basically also is affected by heart, respiration patterns and so on. The scanner and the motion basically also introduce additional noise. So, I will show you the picture of a scanner shortly, and any kind of motion that the body does also introduces additional noise, which needs to be taken care of before you are interpreting data from fMRI. It requires statistical modeling, general linear model, to infer the activation, the cause source of activation and how the activation must be interpreted. Sometimes if you are not very careful with the design of an fMRI experiment, it can lead to false or even misleading calculations. This is typical example of the fMRI scanner. You can see this is the circular magnet where the person is laying, and the magnet basically is, because of the magnetic properties of the blood, it is able to track where the blood flow is more or less in the in the brain, which areas are receiving more oxygenated blood which has ferromagnetic properties, and which areas have received less oxygenated blood. So, that is basically what we are trying to do here. Now, neural activity, wherever the brain is involved in any cognitive function, there is more neural activity, and this neural activity increases the metabolic demands on the brain. So, the brain starts consuming more oxygen. The cerebral blood flow increases and it reaches that area of the brain to provide for more oxygen consumption, which basically reduce to deoxygenated or deoxyhemoglobin in a particular area, which is basically referred to as the blood oxygen and oxygen level dependent response, or the BOLD response, which forms the basis of most of the fMRI studies that you will talk about. Now, as we know, this is an indirect measure. BOLD reflects mostly vascular activation, basically how the blood is flowing through these arteries, and it is not merely a neural signal. It has a larger spatial spread, because the veins are, some of the veins there is a lot of blood flowing up, and some of the veins are draining because the blood flow is moving from one direction of the brain to the other. Also, sometimes these signals may originate away from the actual neurons where the activity is happening, and this is something that we should keep in mind for better interpretation of fMRI data. Now, there is certainly a temporal delay in the development or in the build up of this BOLD response, and this delay is generally to the order of neural events typically happen in milliseconds, but the BOLD onset, the blood oxygen level dependent response, it basically onsets generally around 2 to 4 seconds after the neural activity has happened. It peaks in around 4 to 6 seconds, and it returns to its base in around 12 to 16 seconds. So, you can see the spatial activity in the brain, or the vascular activity in the brain, the blood oxygenated oxygen level dependent response actually builds up much later than the neuronal activity has happened. That is the reason that there is very limited temporal precision. There is a very constricted temporal resolution of this method, but it has a very good spatial resolution, which basically tells you which areas of the brain were involved in performing a specific cognitive function. The basis of how we analyze fMRI data is based on the hemodynamic response function. The hemodynamic response function, contingent to the cognitive operations I will show you very shortly, is slightly delayed, and it rises gradually. It includes peak where it reaches its maximum, and the undershoots when it sort of comes down to rest, and it varies across different brain regions. So, this is also something important that we do not know, that the HRF or the hemodynamic response function in different brain areas will be different, and it makes the interpretation of this data slightly difficult. Canonical HRF is what is typically used in modeling of this data and statistical analysis. So, sometimes there are overlapping HRF functions which create ambiguity of which regions of the brain they are emanating from. Sometimes when the stimuli are placed very closely together, we have seen the time course of the development of the bold response. If the two stimuli are placed very close in time with each other, let us say 2 seconds or 3 seconds, then they will have overlapping HRFs and it will make it difficult and will create ambiguity. Design therefore must, a good experimental design for fMRI must minimize collinearity, that the stimuli of interest are not presented just one after the other with small time in between. They should be actually well spaced out. So, timing optimization in these cases is extremely critical. Now, one of the ways in which we can sort of work out the HRF function is through convolution, which basically combines and overlaps the neural events with HRF. It produces predicted bold signals with in the areas where the brain is active, and it is the one that is used to create regressors that will basically help us model the response or model the HRF function. This is one of the core steps in the generalized linear model that is used to understand or analyze the fMRI data. This is basically the explanation of the HRF. You can see the HRF describes how neural activity is translated into a measurable bold signal. After a neural event, say some event has happened, a face has been seen, the blood flow increases leading to the delayed rise in signal. The response peaks at around 4 to 6 seconds, and then it returns to the baseline. Sometimes an undershoot often follows due to delayed vascular recovery, so that the response is still there. So, this is something that as statisticians, as data analysts, fMRI researchers are actually looking for. Convolution is here, say for example, neural events when they are plotted together, when they are combined with the HRF through convolution, this produces the predicted bold signal using the generalized linear model. So, you can see these blue peaks are where the stimulus is presented, the HRF function is this one, and the bold convolution basically it tells you how the signal is coming in response to the neural activity. That is why experimental designs in fMRI can be sometimes blocked or event related, and we will talk about them. Blocked designs, however, are better as they produce sustained signals with relatively high power. You can see these blue sine curves are the blocked designs which basically create sustained activity in the bold response, and these orange ones are the event related designs which discrete trials are presented with the critical stimuli. So, two types of designs are used: block designs, which basically create sustained activation; event designs, which are event related fMRI designs, which basically involve presentation of stimuli one by one in discrete trials. There are also mixed designs which basically include both sustained and transient sort of build up of activation, and the choice of which design that you want to follow basically depends on the research question that is being attacked in a given fMRI experiment. Now, block design basically has a typical on off condition. The stimulus is present, then there is silent for some period, then the stimulus is presented again, then there is silence for some period, and it is presented again. It typically produces a strong cumulative signal in response to that particular stimulus, and it makes it easier for the researchers to detect the activation, the bold activation in response to that particular stimuli. Block designs also have pros and cons. For example, the very good advantage is that it has high statistical power. It basically allows for robust detection of activation, but in some cases you can see that it is very predictable, and it might have an influence on the way the brain is cognitively processing the stimulus, and it is something that cannot isolate individual trials when specifically in time a particular stimulus was presented. So, that is why to counter that, an event related design was thought of, which basically allows for discrete stimulus presentation. Stimuli are presented in separate trials with sometimes random or jittered timing. Each trial is modeled separately. So, each trial, when the bold activity develops and goes to the baseline, is modeled separately, and it therefore allows trial level analysis. So, when you want to compare different kinds of stimuli within a particular block, the event related design is the one that is more suitable. It also has its pros and cons. For example, the event related designs are more flexible and they are more realistic. They can examine variability of how the brain reacts across trials, but they are basically slight, they are lower sustained activation than block design, and it requires many more trials to actually be able to infer how the brain activity is happening in response to specific stimuli. Now, the mixed design basically has best of both worlds. It combines both block level and event related effects. It captures both sustained attention and transient responses across trials. It is slightly more complex, therefore, more complex modeling is required to analyze data from a mixed fMRI design. Timing is of extreme importance in fMRI design. So, the inter-stimulus interval actually determines whether the responses will be overlapping or not. Typically, it is preferred that the stimuli are evenly spaced and they are very well separated from each other, so that the HRF for different functions are non-overlapping and not creating ambiguity. Short ISIs, if they are there, they will create overlap in the HRFs. Long ISIs will basically sometimes lead to inefficient scanning. So, if the two stimuli are placed farther than 10 milliseconds or 8 milliseconds from each other, then also it will lead to inefficient scanning. So, a balance is absolutely necessary, that you present the stimulus for the exact amount of time so that it leads to the development of the bold response, but it is not so far spaced apart that you cannot actually interpret why and how these effects are building up. Jittering is therefore a very important mechanism that you can jitter or randomize the timing of the stimulus presentation. It basically improves the estimation of the HRF, because one is happening and then after an specified time of amount of time, the other trial is happening, another unspecified time of amount of time, another trial is happening. It basically makes interpretation of the HRF much more easier. It reduces the effects of predictability across participants and across individuals, and therefore it helps in how the individual is processing a given stimulus. Both of these are more efficient, and basically the efficiency of an fMRI design depends on the design matrix. The goal is to basically achieve maximal statistical power, and there is obviously a trade-off between detection of activity and estimation in terms of the neural activity. Now, the overall analysis, and this part I will do very quickly because I am not going to cover the generalized linear model in any detail. They basically model voxel time series. So, the volumetric section of the brain, the voxel, and how the activity is developing over time in that. It uses multiple regressions and bold and basically it tries to predict the blood oxygen level dependent response from the design features. Basically, the idea is that the observed signal, whatever you are getting, part of it will be predicted, part of it will be error due to different reasons. The model is created such that it can estimate and minimize error. A very good fit is basically one that leads to accurate inferences and takes away the noise from the data. Now, the design matrix basically is how the experiment is actually designed. The columns are the regressors which are going to be used in interpreting the data. The rows are the time points where the data is taken from, and it basically encodes how the different experimental conditions are involved in the experiment. The regressors basically are task regressors which are convolved with the HRF. Motion regressors are used to remove artifacts. Drift terms model basically slow and understand the changes. There are also beta weights which basically look at the contribution of each regressor that you are using to estimate how the bold function is actually going on, and this is the one that is absolutely useful for hypothesis testing. Basic analysis in fMRI is basically contrasting the two conditions, the task condition and the baseline condition, and this is basically what is most important. So, this is what is referred to as the subtraction condition. You look at the activity of the brain at rest at the baseline level, and you compare it with the activity of the brain at the time when the task is basically getting performed and the task is going. You subtract that, you get the activity that is contingent to the specific task that you have asked the brain to perform. So, it basically is a extremely important aspect of the analysis. Then you get your statistical maps. Each voxel is analyzed independently. It produces statistical map of activity throughout the brain, and it can be visualized as the brain activation during the particular task or the baseline. There are multiple comparisons of these kinds possible, thousands of voxels are there to test. It is, and because there are thousands of these comparisons possible, there is a high chance of type 1 errors or false positives, something that we were talking about in ERP data analysis as well. So, it requires a lot of cleaning, it requires a lot of correction. There are different kinds of correction methods, you can have different kinds of cluster correction using spatial extent and so on. I am not going into a lot of detail of this because we are getting into the analysis part, but the idea is that this data really, it's a lot of data, it's rich data, and it needs to be cleaned very carefully in order to reach the correct inferences. There is a pre-processing protocol there. So, raw data contains obviously noise. Pre-processing, removing the initial noise will improve quality. It is definitely essential before you push the data into analysis. And the analysis pipeline, you will have to have slice timings, which areas, which time periods of the brain you are basically acquiring. You have to use motion correction, realignment. So, any sources of motion have to be taken out. Normalization of the standard brain, basically smoothing also improves this function. You have to remove motion related artifacts, correct via realignment, and these motion parameters are added to the GLM so that it takes care of them while interpreting the overall thing. Typically, one of the very important steps is normalization. It maps the entire brain to a standard standard space. It allows for comparison between participants and allows for comparison between groups, and it also enables averaging across subjects so that you know that okay, voxel A, B, C is the same as voxel D, E, F in subject 1 versus subject 2, and you can actually compare the activities in these areas. Smoothing is required to sort of take away the extra noise, improves the signal to noise ratio, but it reduces spatial precision in terms of which specific areas the activity was arising from. Obviously, there are several sources of noise. So, heartbeat, respiration, scanner drifts, subject's motion artifacts. So, all of these are there, which as I have said, you have to be taken care of. Now, interpretation of fMRI is extremely important. One needs to understand that fMRI basically shows correlation data and not causation data. This is something I was mentioning in the previous lecture also, and activation does not involve the necessity of that area. If a particular area of the brain is active during a particular cognitive function, it does not imply that that area is absolutely necessary for that cognitive function to be performed. Therefore, a very careful interpretation of fMRI data is required. The fundamental rule that one has to remember is fMRI is correlational data. It is not causal data, as to it does not really tell definitely that this area is definitely involved in this particular cognitive function. Obviously, there are some limitations of the method. For example, as I said, the bolt signal takes time to develop, and therefore it has low temporal resolution. It is an indirect metabolic measure, so it is not direct neuronal activity that we are measuring, and it is obviously sensitive to vascular defects, defects in patterns of blood flow and so on. So, these are some of the things that I sort of wanted to talk about. There are obviously best practices that you have to optimize your design before scanning, you have to control for confounds. Everything that we have talked about in experimental methods so far is something that you have to take care of in any of these experimental methods that you are using, whether it is eye tracking, whether it is ERP, or whether it is these neuroimaging methods. Use proper corrections, control confounds, and obviously most important of all, avoid over interpretation. So, these are some of the things that I wanted to talk about, and I hope this course has been useful for all of you, and you have learned some of the critical aspects of experimental design and a little bit of a overview of different experimental methods that are used. Thank you. I hope this course will be useful for everybody.